So today we're going to talk about cell replication and patterns of inheritance. The core concept here are that the development of biological organisms depends on information flow from genes, which are encoded in DNA, to RNA and protein. That used to be called the central dogma, but we now know that viruses do different things, so it's not as central anymore. It's central for us, but not so much for some other organisms, some of which may not be called alive, but that's neither here nor there. So organisms inherit genetic and epigenetic information that influences the location, the timing, and the degree of gene expression, and that's what we're gonna talk about. This genetic information is transmitted to the offspring via cell-specific regulated cell reproduction, and that's the other part we're going to be talking about. So to talk about this, we need to talk about cells. Cells are the basic units of life. They've been called the atoms of life because they're the smallest self-replicating unit. Cells don't come to start de novo. A cell comes from a cell. Human cells are diploid. That means they have 2 copies of the genetic material in every single human cell. So when a cell replicates, it needs to divide genetic material into the 2 cells. And there's 2 ways how a cell go about this, depending on what the purpose of it is. There's, on the one hand, there's the somatic cell division, and then there's the reproductive cell formation. Somatic cell division happens when cells, like if you get injury on your arm and your skin cells need to replicate and make more skin cells to fill in the hole, basically. In that case, somatic cells, which is the body cells, they undergo mitosis, which is a replication and duplication of the DNA, so that you have 2 cells at the end that are exactly identical to the initial cell. The reproductive cells are formed by meiosis. In the case of meiosis, the diploid original cell is going to be split in such a way that you end up with a single copy of the genetic material. And that happens in humans in the egg and the sperm cell, because in order to create offspring, you need an egg and a sperm cell that go back together. So in order for them to be diploid, they need to be haploid, which is what it's called when you have half the genetic material to start with. Now, when you're looking at the cell cycle, when people first started looking at cells under the microscope, one of the things they noticed is that they looked different, right? There were some cells that looked kind of like this, and some cells that looked kind of like this, with like these stringy things, and some of them were curled up like that. And so people were looking at this going, that is rather curious. And so they decided that where this stuff is happening here, we have all these stringy things, where the action is. Whereas all of this, that's the in-between. That's when nothing is happening. So they called this section where nothing, in quotation marks, was happening, the interface. Inter means in between. That's the in between the action phase. So now we know that the interface is anything but nothing is happening. A lot of things are happening in the interface. And the place where this action is happening, where actually cell division is happening, which is mitosis and cytokinesis, are very, very small parts of the cycle. Most of the action is actually happening in the interface. And when you look at the interface as a phase, you've got G1, so you've got S, you've got G1 and G2 and M. G1 is a checkpoint where, so there's 3 major checkpoints. One point is G1, That's the checkpoint to make sure that DNA synthesis can begin, that everything is in order, everything is there that we need. G2 checks if DNA synthesis has been completed correctly, and there's where the commitment to mitosis happens. Once that checkpoint is completed, mitosis is happening. And then M is a checkpoint where it checks whether all the chromosomes are attached to the spindle and if the sister chromatids can be separated correctly. This progression is highly, highly conserved. And so when we're looking at the phases of mitosis, and you've probably seen this before, but before I go into the details of this, I actually want you to think in terms of the logical things that need to be accomplished during mitosis. And for that, I have a little mitosis a la Stephanie PowerPoint. So if you think about this, because I don't want you to memorize, I want you to memorize, but I don't want you to memorize the phases as just phases. I want you to think about what needs to be happening there. So if you start out with a cell, and it's got 3 chromosomes here, a red one, a green one, and a blue one, just for sake of argument. And what we want to end up with in the end is 2 of these, right? We want to turn 1 of these into 2 of these. So to do that, we start with 3 chromosomes, we end up with 6. We need to start by duplicating these chromosomes, right? So here we go. We're going to duplicate these chromosomes. Now they're duplicated, and so all we have to do, obviously, is just split this cell in half, and off we go with 2 new cells. When I draw this in the class, at this, I usually ask my class, I'm like, so, you know, we're good to go, right? And somebody usually raises their hand and says, yeah, no, not so much, right? Because these 2 resulting cells are not the same as the original cells. And so you got to think about these. This is chemistry that is happening here. There are molecules, macromolecules, and we've talked about macromolecules already, that somehow need to make this happen, right? So this isn't it. So let's go back to where we were. But we do need to duplicate the chromosomes. But when we do, we need to keep track of where they are. And the easiest way to do this is when you're duplicating this thing anyway, is you just keep a hold of it somehow, right? And so these chromatids, there's 2 of them that we end up with, and they're just kind of attached together. Now, the next thing that happens is, and again, in the classroom, I liken this to the picking teams, because what you're going to have to do is you're going to have to make sure that each team gets 1 of each players. And the way we do this in the playground is we line everybody up who's selectable, and then we've got 2 team captains who are like, I'll pick this one, I'll pick this one. And then a lot of times I make fun of, you know, current students who probably don't play outside and all of that. But either way, so the next thing that happens is we're going to line them up in the middle. And we're going to make sure that, you know, those 2 captains to the right and to the left, they can basically put out their, you know, strings, their fishing poles, they're not fishing poles, but fishing lines, to grab a hold of the player they want. And the next thing that happens is these selectors there get a hold of the people they want, or the chromosomes they want, and they pull them towards each other. Now, for sake of brevity, I'm sort of skipping the details over here, but then once you take the chromosomes to the side that you want, you can then start dividing the cell. And this is exactly what's happening there. And then you can have 2 cells that are divided into each other. So this is the logic behind what's happening. And so now that we have an idea of the logic behind what's happening, let's look at these phases again and see if they make a little more sense now. So we're back to mitosis, which is the equal division. So let's start putting names here together. So in the prophase, you've got the chromosomes that condense. There's these fibers, these spindle fibers, which are the selectors, basically. And they move to the opposite end of the poles because they're going to have to be doing the selections. In the prometaphase, the nuclear envelope disappears. The chromosomes condense. They're at this point they're duplicated and attached to each other, and they're keeping track of each other. In the metaphase, meta means middle. In the metaphase, they're lining up in the middle, and they're, the poles are there. And from each pole, there's a fiber that's being attached to each chromosome. The anaphase, ana means separate and back. It means a couple of different things, but think in terms of analysis. Analysis is when you're pulling things apart, right? Because you're analyzing things, you're trying to figure things out. And that's when things get pulled apart to the poles. In the telophase, telos means end. And in the telophase, you have the envelope gets rebuilt, the chromosomes kind of get back into their original shape, and then in the cytokinesis, the whole thing gets pulled apart. Now I've got in this PowerPoint, I've got pictures of what this looks like in detail, and I want you to kind of know what's happening in each phase. So you may be faced in the exam, you may be faced with one of these pictures like this one, and then you may find an error without the label that says, what's this? Or, what's that? So in the prophase, you've got the separating centrosomes, you've got the nuclear envelope that's disappearing, the chromosomes that are condensed. In the metaphase, you've got the chromosomes in the middle. The kinetochore is where this microtubule attaches. Centrosomes are also called spindle poles. In the anaphase, you've got the separating chromosomes. Did I just skip telophase? Yeah, I skipped telophase. In telophase, you've got the reforming of the nuclear envelope, and then you've got cytokinesis. Now, cytokinesis, we're going to look at two types of different cells today. We're going to look at an animal cell, and we're going to look at a plant cell. The animal cell has a cleavage furrow, which is right there where the contractile ring is at. Whereas, so the animal cell kind of pinches in the middle. That's how the animal cell separates in cytokinesis. Whereas the plant cell is different. The plant cell builds a cell wall. And that has to do in part because plant cells have cell walls. Animal cells don't. Plant cells have rigid cell walls, which actually, and we talked about, I think it's the previous class, we talked about osmosis. Plant cells are very resistant to being filled up with water. They need to be filled up with water. And part of what makes that possible for them is the cell wall. So obviously with the division, you also have to be looking at them a little differently. What you're going to be looking at in class is you're going to be looking at, you'd be looking at that under the microscope if we were in actual class. Since we don't, you're going to be looking at pictures. And what you're going to be looking at is you're going to look at allium, which is onion root tip, where you can see, because these chromosomes are stained, where you can see the different phases. And what we're going to be doing in class, we're going to be figuring out how long each of these phases lasts. So how long the inner phase is and how long the different phases in mitosis are. Now the way these look is you've got a prophase, which is the curled ups of the of the chromosomes. Here's an anaphase where they've been pulled apart. Metaphase, they're in the middle. This is another metaphase. This is an anaphase. This is a telophase, and this is an onion root tip, so that's a plant, right? And so you can see a little bit here of the cell plate that's visible. This is an interphase, which means that's not nothing in quotation marks happening. This is another anaphase, and you'll see lots of these cells. Because you're going to have to count like 100 of them. So the next one we're going to look at is the whitefish blastula. In real life, this is much harder to see because they tend to be very, very faint. But basically, this is what an interphase looks like. The anaphase, there are the chromosomes that are being pulled apart. Here's another anaphase. Here's a metaphase where the chromosomes are in the middle. Prophase, where you see a little more pronounced chromosomes. And then this one's a telophase because you see this invagination, this cleavage furrow right here. Now, when we're going to be looking at the timing of the mitotic cycle, mathematically, it's a very simple thing to do. Basically, what you do is you're going to say, well, so the basic gist of what you're doing is you're going to say the entire cell cycle of N with allium takes 16 hours. So because when you cut through the onion root tip, that is a moment in time. And so each of these cells is caught at a specific moment in time in its cycle. So each cell goes through the cycle, and each cell is going through a moment in time. And then by taking a snapshot of where everybody is in this time, that's a reflection of what percentage each cell spends in that particular phase. Now, the next slide is going to make you be cross-eyed, so be prepared for that. The next slide is an attempt by me to try to explain to you what this moment in time, how this actually reflects your ability to see how much time each item spends in each phase. So don't go cross-eyed here. So you've got what you've got here is you've got 28 different supposed cells, right? And they go through phases. So there's a yellow phase, a green phase, a blue phase, a red phase, an orange phase. And it's a regular phase. So there's one of the yellows, and each cell goes through the same cycle, but they're not going through the same cycle in the same order. They're going through them in a different order. But this is the time, this is the direction of time. And so time goes this way, right? And so the idea is if I now cut this, like here, for example, at a specific time point, then there's going to be cells at different phases, and where everybody is will reflect the overall time frame. So, and the next slide shows you this kind of hopefully a little smaller. So this is the real cycle. So you've got 4 reds, and each of them is going through that same thing. You've got 4 reds, 3 oranges, 1 yellow, 2 greens, and 2 blues. So everybody's going through this. And like, let's take this snapshot in time of 28 of these cells. And if you count up how many red ones, how many orange ones, how many yellow ones, how many green ones, and how many blue ones you have, you can then say, okay, so out of 28 cells, 11 were red, 7 were orange, 2 were yellow, 4 were green, and 4 were blue. Now, if 12 hours is our total time, so 100% of the cells is 28, then 11 reflects 39.3%, 7 reflects 25%, 2 reflects 7, 4 reflects 14.3, 4 reflects 14.3. And if the overall cycle is then 12 hours, which is the same number as 1 through 12 over here. Then the red phase, based on calculation, reflects 4.7 hours. The orange phase would be 3. The yellow phase would be 1. The green phase would be 1.7 or 2. And the blue phase would be 1.7 or 2. Now, obviously, these numbers are not exact, but the more numbers you would have here, the closer this number would come to reality, because the odds are that you get the numbers that you need. If you look right and left, this is the same calculation, so you could do the same calculation at each time point. If you don't understand this, don't worry about it. This was an attempt to try to clarify something. We're just going to do the calculation. So if you just believe me for the demo part, just believe me for the demo part. So, but those of you who want to like logically try to figure this out, this is a way for you to logically look through it. The next thing we're going to do, so this is cell cycle, and this is mitosis and the timing of alum. And this is going to be in your handout on pages 2, specifically page 4 is the timing of the mitotic cycle, and I'll go through that in detail with you guys in the demo part. But the next thing we're going to have to talk about is basic principles of inheritance, and we can't get around Gregor Mendel when we do this. So Gregor Mendel was actually a monk and a mathematician, and he mathematically figured out how things are inherited based on math. He used the flower of peas, and they didn't understand chromosomes yet at that time. They didn't know how all of this happens. But he said, all right, there's seems to be these 2 factors that he called alleles. And there seems to be 2 parents who bring 2 of these alleles, 1 of each, to the offspring. And this seems to be independent. They seem to be separated from each parent, and they seem to be independent. So this is how he came up with the law of segregation and the law of independent assortment. This is true. What Mendel figured out actually very much reflects how chromosomes work. What Mendel didn't realize was that sometimes some traits have inputs from many genes. And so we talk about non-Mendelian genetics, which I think is unfortunate. But so what he said, so we're going to have to learn a couple of terms now. So the observable, measurable characteristic of a gene, of an appearance, is referred to as the phenotype. So what it looks like, that's the phenotype. So blonde hair, tall, freckles, whatever. The genetic makeup that is responsible for this phenotype is the genotype. Those are the underlying genes. A phenotype is usually a word. A genotype is always 2 letters. Well, if it's 1 gene, it's 2 letters, because we're diploid, so you need 2 letters for this. Alleles are the individual letters. So those are the alternate versions of the gene that produces those different variations. Now, you can either have the same of these 2 genes or 2 different ones of those 2 genes, right? Of those 2 alleles. If you're heterozygous or heteroallelic, you have 2 different alleles for a specific trait. For example, TT would be if T is tall and little t is short, that would be heterozygous. Homozygous would be they're the same. Now, in Mendelian genes, those are the simple ones that I inherited. So where just a simple gene, 1 gene determines the outcome. So So the dominant allele here is always symbolized by a capital letter. There are reasons why something is dominant and why something is recessive. But for your purposes, you really just need to know that the dominant allele is always symbolized by an uppercase letter, whereas the recessive allele is always symbolized by a lowercase letter. Now, one of the things I want you to know about dominant and recessive, dominant does not mean it's better. Dominant just means it's the one that's visible. If you have two of them, the dominant ones is visible even if the other one is the recessive one. Whereas for the recessive one to be visible, both alleles need to be recessive. If there are more than one gene involved, or sometimes you have things that are not, if it's not a dominant recessive kind of situation, and we're going to play through some of those in the lab today, it's called non-Mendelian. It does not mean it's not chromosomal. It's the same thing. And Mendel would have probably, had he known about chromosomes, but he didn't, had he known about chromosomes, he would have probably adjusted his the way he described these things. So when people figured out that how mitosis and meiosis were happening, when they first started looking at that under the microscope, they rediscovered what had been forgotten in time. It happened really very closely to each other in history. But Mendel was in Germany. Well, so was the guy who figured out that how mitosis happened. The first guy ever describing mitosis was also in Germany. But it was a bunch of Americans. Actual Thomas Hunt Morgan, who was born in Lexington, Kentucky, who figured out how these things went together. When you look at the human chromosomes, you can make this thing that's called a karyotype, which lists all the chromosomes we've got. We've got 22 plus the sex chromosomes. And the karyotype, we use it actually in prenatal tests, in amniocentesis, where we're going to be looking at whether mostly for Down syndrome. And then we have another one that's the chorionic villus sample that's also being done. And it can be done earlier than amniocentesis. So if we're thinking something's wrong, sometimes we'll do the chorionic villi sampling. But the way that the formation of gametes happens is with meiosis. Meiosis is, so where mitosis creates a nearly identical copy of two cells, meiosis promotes genetic diversity. And especially that happens in two different places. One in the prophase, where you can have crossing over. So chromosomes are kind of floppy, and as they're being duplicated, they can be really close to each other, and sometimes they like break at the same place, and then you end up with like a piece from the other one. This, if it doesn't work out, then it just doesn't replicate right. But if it does work out, then all of a sudden somebody's got brown eyes and freckles rather than blue eyes and freckles. The other source of the variation is in the independent assortment, where it's where there's no rule that like the genes from the father will go to the son and the genes from the mother will go to the daughter. So over here you see an image of this where whether the blue one or the red one goes to the spindle pole one way or the other is completely independent. And with the number of chromosomes we have, the variation is just about endless. And then of course the other part of the variation is mate selection, right? It makes a difference whether you pick somebody who's got blue eyes or brown eyes. And maybe we'll talk about a little bit of that later. But so similar to mitosis, meiosis has different phases, and I really just want you to basically know the overview of this. And it's essentially, you can almost think of it as a two mitoses in a row. But initially, you're lining up, instead of lining up two chromatids, you're lining up two chromosomes, and then you're pulling sets to the side, and then you're making, after the first telokinase and cytokinesis, you're going through prophase 2, metaphase 2, anaphase 2. So basically, this is another mitosis that happens here that ends up with like daughter cells with individual haploid a haploid set of chromosomes. So this is how gametes are produced, right? You have meiosis, you have the first meiotic division, and then you have the second meiotic division, and you end up with a genetically diverse set of gametes, each of which are haploid. And that is all I wanted to tell you for this part, and then you'll go on to the lab part next.